Well, good morning, everyone. Good day. Welcome again from Stanford with the EMX seminar series. Today is the 60th day of the 2021, and we're on our third seminar already. So that's been very exciting, and we appreciate everyone's participation. So naturally, we have two exciting talks today, one from the United States and one from Sweden. And so then I will hand over to Wachu, who will then introduce Bryan, first speaker. Thank you.
Hello, it's my great honor to introduce my colleague, Professor Bryan Kobielke from the Stanford University Medical School as our first speaker today. Bryan is very well known for his structural work on G-protein coupled receptors, which formulate a family of proteins involving in many key biological functions so that we can see, smell, and taste and feel. So his extraordinary innovative work has won him the Nobel Prize in 2012 in chemistry, together with his former mentor, Robert Lefkowitz at Duke Universities. Bryan has been using both X-ray crystallography and cryo-electron microscopy to determine the structures of many G-coupled protein receptors. Today, he will share with us his structural insights in this very important membrane protein, which has been a major drug target in multiple diseases. Bryan, welcome.
Thank you. Okay, can you see my screen?
Yes.
All right, welcome everybody. Good morning to those of you who are on the West Coast, and good day to the rest of you. So I've been asked to give an overview of the impact that cryo-EM has had on our field, G-protein coupled receptors. So first I'm going to give you a little overview of what receptors are for those of you who aren't in the field, and then a little tour through what we've learned about them through initially crystallography, and more recently cryo-electron microscopy has really become an extraordinary tool for studying GPCR structure and function. G-protein coupled receptors mediate most of the hormone and neurotransmitter signaling in the human body. They also mediate the responses to sight, smell, and taste. The general mechanism of G-protein coupled receptors is shown below. The receptor shown in green, when activated by an agonist, will activate heterotrimeric G proteins composed of an alpha, beta, and gamma subunit. When activated, the G protein dissociates GDP and then binds GTP, which leads to dissociation of the trimer into a free alpha subunit and a beta gamma subunit. And both of these subunits can act as signaling proteins to modify the behavior of either channels or enzymes in the cell. Upon hydrolysis of GTP, the trimer reassembles and the cycle can begin again. So there are over 800 members of this very large family, which is shown here. And shown in the upper right are the olfactory receptors. So the majority of our GPCRs are dedicated to sensing odorants. And this number is even larger, for example, in animals that rely more heavily on odor detection for finding food or avoiding being food. For example, mice have roughly 1,000 olfactory receptors. There are also 4 receptors dedicated to vision. These 2 clusters reside in the largest subfamily. So family A composes about 700 of the 800 members. And they include receptors for things you might have heard of, including adrenaline, acetylcholine, dopamine, serotonin. Also receptors for opiates, cannabis. So these are all family A GPCRs. We also work on two other family, including the secretin family, and these are important for carbohydrate metabolism, targets for drugs for diabetes. And then the other family, the glutamate receptor family, also called family C, and these are neurotransmitter receptors that are important in behavior. So because they're the largest group of receptors for hormones and neurotransmitters, they're the single largest class of targets for the pharmaceutical industry. So when one wants to understand how a biological process works, structures are really important. So from my time as a fellow in the Lefkowitz lab through my career at Stanford, I've been focused largely on getting structural insights into what receptors look like and how they engage G proteins and other signaling molecules. However, the main problem has been that GPCRs are highly dynamic. It makes them very challenging subjects for structural studies. So the first part of my talk, I'm going to briefly review what we've learned from crystallography and then how cryo-electron microscopy has really expanded our tools and enabled us to get structures much more readily. And then also just mention that in addition to trying to understand how receptors work in molecular detail, cryo-electron microscopy has now really enabled structure-guided drug development for G-protein coupled receptors. So on the left here is just a characteristic of all GPCRs, and an important consideration for drug discovery is that when you develop or when you're finding a drug for a particular GPCR, it's not just an activator or an inhibitor. Drugs have a really wide range of effects on receptor activity. So the biological response is on the left, the drug concentration is in the x-axis, and you can have drugs that are full agonists, and typically these are similar to the native hormones and neurotransmitters. Then you have drugs that have a range of activity called partial agonists that just either have a weak or strong activity. And most of the drugs that are activators are partial agonists. They tend to be safer and better tolerated. Receptors also have a certain level of basal activity, and many of them do anyway. And this can be suppressed by drugs called inverse agonists. So what kind of simple model can explain all this behavior? Well, for the longest time, we used a two-state model for receptor activation, where receptors exist in equilibrium between inactive and active states, and that the G protein recognizes and stabilizes the active state. However, we now know that this simple two-state model is far from being correct. Receptors actually, we now know, exist in an ensemble of conformations, and this data comes from deer and NMR studies, and sorry. I'm sorry, this comes from deer and NMR studies, as well as C13 methionine NMR studies, where we can see receptors exist even in completely unliganded receptors, an ensemble of conformations. And some of these conformations can be nearly active-like conformations as a result, resulting in this basal activity that we see. Agonists don't actually stabilize a fully active state. They just increase the probability that the receptor will exist in an active conformation. And only when the receptor is coupled to a G protein or other kind of signaling proteins will it be in a fully active conformation. So this is a molecular dynamic simulation of the beta-2 adenergic receptor, the one we spent most of our time working on. And it just gives you a sense of the dynamic character of these proteins. Now, so what are the challenges for getting structures by crystallography? Well, clearly protein dynamics is incredibly important. And what that means is that receptors exist as a in basal state as a heterogeneous population of minor conformations. The other is that there's relatively little polar surface for crystal lattice contacts. So when we were trying to figure out how to get around these problems, we chose two different approaches. One was to identify antibodies that might recognize the most dynamic components and stabilize them, and also provide polar surface area. And the other was to do this by protein engineering. And this was accomplished, both methods ended up working. In 2007, we were able to publish the first structures of the beta-2 Adenergic receptor. The method shown here with the insertion of T4 lysozyme, which is a highly crystallizable soluble protein. This general approach has been used to crystallize most other family A GPCRs. The proteins that have been inserted are sometimes different, but the general principle is the same. So we now had some components of this signaling cascade identified. We had inactive structures using a variety of different methods. We then tried to get structure of an active state. And we even generated a covalent agonist to stabilize the active state. We're able to get a structure again with fusion protein. However, the structure was in exactly the same conformation as the inactive state. So even though it was bound to a covalent agonist, it stabilized, it crystallized as an inactive conformation. So this brings us back to our dynamic model in that we found from using other methods that receptors, when bound to an agonist, still predominantly exist in inactive states, and agonists only increase the probability of being in a more active conformation, and that we needed something to stabilize it. Well, we were trying very hard to stabilize the complex with G proteins, but we also went back to using antibodies. And this was a really fortuitous collaboration with Jan Ster, who I met at a Gordon conference, who had been developing camelid antibodies, antibody fragments called nanobodies, as protein stabilizers. And camelids have a class of antibodies which are different from ours, shown on the left, which are heavy chain only, and therefore the variable fragment is smaller, oval shaped, and more effective at stabilizing proteins because they can insert themselves into crevices, whereas fab fragments tend to bind to flatter interfaces. So shortly after sending in protein, we got antibodies back, antibodies, and we're able to obtain the first structure, active state structure of the receptor. So we now had a picture of what the receptor might look like before engaging G protein. And however, our goal was to be able to get the full receptor G protein complex. And I'm going to spare you a lot of the details. This took a lot of trial and error and a long time, but just to give you an idea of some of the challenges, all of these things were necessary to enable the crystallography, and it involved expertise from a variety of different individuals. We had to find a super high affinity agonist that would stabilize the complex, and we screened compounds from 3 different pharmaceutical companies, a total of about 60 screened, and found an agonist that had an extremely slow dissociation rate. My colleague Roger Sunahara determined that the G protein must be nucleotide free to be stable. We had detergents that were synthesized by a chemist that weren't commercially available, that were important. We had lipids that were synthesized by a lipid chemist, also not commercially available. We ended up having an antibody that helped to stabilize, and we did protein engineering with an amino-terminal T4 lysozyme. And then finally, even back then before cryo, Yorgos Skiniotis really provided a lot of useful information by negative stain cryo-EM. And what he found was that that the whole complex was also very dynamic, and that this particular component, what we call the alpha helical domain, was flexible and exists in a variety of different conformations, and that this was primarily the reason we weren't able to get crystals. And using negative stain, he found a specific nanobody that helped to to some extent. So this is our trip back from that synchrotron, and these are the individuals who played the major role in getting these crystals. So we now had finally our first receptor G protein complex, and this is again just shows all the extras we had. We had an amino terminal T4 lysozyme, and then this brown, this green is the nanobody, the brown and gray are the beta gamma subunits, and the gold is the alpha subunit. So briefly, what did we learn? Receptors and G proteins engage as shown here. I'm going to go back and forth between inactive and active states. And you can see the major changes in the receptor, actually, there are relatively small changes where the agonist binds. And this propagates down into a very large structural change in the transmembrane domain 6. This opens up a cavity in the core of the receptor that can, what we call the alpha-5 helix of the G protein engages. And that leads to dissociation of the GDP. And you can see this very large movement of the domain down here, the alpha helical domain. So we now have one structure of one receptor coupled to one G protein subtype. However, as you can see, there are a number of different G protein subtypes. We are receptor coupled to GS. However, there are family A receptors that couple to GI, GQ, and other, these are the major families. And we weren't really sure whether what we observed for the beta-2 receptor would apply to other GPCRs. So we began to accumulate some other receptors that we thought were particularly of clinical interest and interest for a number of other reasons. And we were able to develop from these 6 different GPCR G protein complexes, so different receptors, different G protein subtypes. They were all very stable, but with one exception, we had no crystals. One, we were able to get really beautiful looking crystals, but they had no diffraction at all. So this is from 2012 to 2017, and now the advent of cryo-electron microscopy. So the challenges with crystallography is that no matter how good your biochemistry is, you need an element of luck in order to be able to grow crystals. But with cryo-electron microscopy, as you know, we just need a nice biochemical prep and the ability to have them stable in a thin layer of vitreous ice. And for most proteins that we work, this is something that we are able to achieve. And as a result, we've been able to get structures of all of these complexes, and a few more are not shown here, just through cryo-electron microscopy in a very short period of time. And these have all been done with the collaboration of Yorgos Kiniotis here at Stanford. Now, I'll briefly mention what we've learned from these, but in addition to being able to get structures of different receptor G protein complexes, cryo-electron microscopy has also allowed us to get structures of receptors with different ligands. Now, this may seem obvious, but by crystallography, often only the highest affinity, most stable ligand would yield to crystallography. So none of these compounds were we able to crystallize the mu-opiate receptor with even with the aid of nanobodies. And we are now able to get structures depending upon microscope availability. So if we have a new ligand and we want to get a new structure, we can get one typically within 4 weeks, sometimes less. And this has been particularly interesting or important for our project on opiate receptors, where we're trying to understand how some drugs that activate opiate receptors are relatively safe, such as this drug called metragenine pseudoindoxanol, and some are more lethal, such as lofentanyl, and cause lethal respiratory suppression. And we're now beginning to see how these different drugs engage different components of the agonist binding pocket. So in general, what have we learned about family A? So nucleotide-free GPCR G protein complexes are similar for all family A GPCRs. The structural changes upon activation are very similar for the G proteins, GS, GIO, and GQ11 subtypes. There tends to be a smaller outward movement of transmembrane 6 for certain GI-GO complexes, and that may be because the C-terminal part of this G protein is slightly smaller. However, the mechanism, none of these complexes really explain why some GPCRs prefer some specific G protein. So the mechanism for complex selectivity and specificity is still really not well understood. And for a number of reasons, we suspect that our structures are at the final stage of this complex formation, this nucleotide-free stage, and that in fact there may be more intermediate states. So we suspect in fact that there is at least one intermediate, and that intermediate is transient and nevertheless may be important for selectivity determination. And evidence for this comes from 3 studies. One, single molecule studies, which I won't talk about today, where we actually observe evidence for an intermediate state that's coupled to the G protein. We've also done hydrogen deuterium exchange and radiolytic footprinting studies where we also observe evidence for an intermediate state. And then finally, by cryo-electron microscopy, we've captured one receptor G protein complex in 2 different conformations. And that's shown here. So this is the neurotensin receptor. It's a promiscuous receptor that couples to GI proteins, GS and GQ11 and arrestins. And when we prepared our preparation for cryo-EM, we observed 2 different conformers in roughly equal amounts. What we call a canonical state that looks like the complex is formed with all the other family A members we have obtained, and a non-canonical state. And the difference is better seen. So on this panel, we're rotating the receptor complex so that we're looking down from the top of the receptor, and we can see the orientation of the G protein. And this dotted line is tracing the alpha N helix of the G protein. So here's the receptor again and the alpha N helix for the canonical state. And you can see for the non-canonical state, it's shifted by about 45 degrees on the receptor. So we then wanted to see whether this might have any functional significance. So we mutated residues that are only observed in the canonical interaction. So these residues are not interacting with G protein in the canonical state. And we then made mutations, and we found that those mutations only affected coupling to GI or GO proteins. So these are closely related proteins. The blue indicates the mutations having a reduced effect following activation. And the GQ, the GI, the G13, and the GS mutations had little or no effect. So this suggests that this state might be important for coupling to GI proteins. We also observed that the alternate state had only one of 2 common structural changes associated with activation. So the first structural change is this outward movement of TM6 that you see in the red and blue conformations, and the other is a unraveling of the helix at the end of transmembrane 7. And you can see here the blue. This is a tyrosine. The blue is the active light conformation, and the gray and the red are the inactive light conformations. So the canonical state has both in the active conformation. The alternate state only had TM6 in the active conformation, but the TM7 state is in the inactive state. So it's partially activated. In the another important component of the G protein is this beta 6-5 beta alpha 5 loop shown on the bottom here. Sorry. And in the canonical state, it's highly dynamic. And in the non-canonical state, it's more stable and largely in a conformation that won't bind G protein. So these are the two loops. So we think that in our model now is that the alternate state is where GDP is released, but GDP can't bind until it rotates into the canonical state, where the more dynamic loop allows formation of a structure which can bind GTP and dissociation of the complex. So that's family A. Now, what about some of these other families? Family B are a smaller subfamily, and they include receptors for glucagon and GLP receptor, which is a target for diabetes medicines. And these family B have a particular structural motif, an N-terminal domain that binds the C-terminus of the agonist peptide, and the N-terminal part of the peptide penetrates into the core of the receptor and activates it. So we were able to get a structure of the glucagon receptor, and it's shown here. It looks very similar to the beta-2 adenergic receptor of family A coupled to the same G protein. So here's the beta-2 receptor, here's the glucagon receptor. The G protein also engages the core of the protein receptor. TM6 also moves outward. There's one major difference, though, is that the movement of TM6 is much more dramatic. So you can see in activation of the beta-2, TM6 is a sort of gradual outward bending, whereas in the glucagon receptor, we have this really dramatic bend in the helix and a disruption of the helix, of the N-terminal part of the helix. And that's shown a little bit more detail here. So the helix bends and it rotates about 180 degrees. So these are incredibly large structural changes and energetically unfavorable. So based on the structure, we began to wonder whether this receptor was less efficient at activating G proteins than a family A receptor. And briefly, it's much less efficient. So we then carried out some studies to look at GTP hydrolysis and GDP release. So the beta-2 receptor shown in this purple releases, the G protein only is shown in purple. Beta-2 receptor releases GDP very rapidly, and the glucagon receptor is roughly 20 times slower. And it also hydrolyzes GDP even at a slower rate. And this isn't because it doesn't form a complex very rapidly. So complex formation is shown here by FRET, and you can see the receptor and G protein engage quite quickly, but the G protein is not able to activate the receptor as readily. And another interesting feature shown here by these fluorescence studies. So this monitors outward movement of TM6 in the beta-2 receptor in the absence of agonist. In the presence of agonist, we see a decrease in fluorescence associated with this outward movement and a further decrease when we add the G protein. For the glucagon receptor, there is no outward movement of the TM6 in the absence of G protein. You need both agonist and G protein before you see this outward movement. And shown here below is that once activated, the beta-2 receptor, when G protein is released, returns rapidly to its inactive conformation. In contrast, when the G protein is released from the GCGR receptor, it is very slow. So this is after several minutes, and it still is largely in an active conformation. So the cryo-EM studies led us to investigate this further and reveal a very striking difference between family A and family B, which is shown here. Family A, you can see some outward movement of TM6 with agonist alone. You see further engagement and outward movement with receptor and G protein complex. Efficient release of GDP upon complex formation. And once GTP binds and G protein associates, the receptor rapidly returns to an inactive state. In contrast, agonist alone doesn't cause an outward movement of TM6, You need G protein, but this happens very slowly, and GDP release is much more occurs more slowly than with family A. And when the G protein dissociates, the receptor remains in an active light conformation for many minutes, in contrast to the beta-2 receptor. So that's a quick tour through family B, and I'm just going to talk briefly about one other family, the glutamate receptor family, which is even more strikingly different in terms of its mechanism. And these receptors exist as a small cluster that include a number of neurotransmitter receptors, including several glutamate receptors and GABA receptor. There are also receptors for calcium and a couple taste receptors as well are in this family. Unlike family A, which are monomers, family B or family C are obligate dimers, and they have this very large extracellular domain, really ideal candidates for cryo-electron microscopy. It had not been possible to get crystal structures of an intact family C receptor, either inactive or active state, because of the relatively flexible connection between these two. So upon activation, the ligand binds to these large extracellular domains, and that leads to a rearrangement of the transmembrane domains. And we were interested in finding out what that rearrangement was. So in order to get the fully active state, we had to use several different ligands, and I just show the functions over here. I won't discuss them, but we used a full agonist. We also used a positive allosteric modulator that binds in the transmembrane domains. And then we have a nanobody that is also an allosteric modulator that stabilized yet again the fully active conformation. And all of these were used to enable us to see both inactive and active states. So shown above is the structure of the inactive state. This is a lower resolution structure, only 4 angstroms. But you can see that the fly traps are bond together. There's a disulfide bond that stabilizes this interaction. And you can see that the two transmembrane domains are well separated. In the active state, the fly traps are closed, and the transmembrane domains approach each other. So this is going to be a short movie, and you'll see the core difference between the active and inactive fly traps. So you can see binding of the agonist shown in purple causes the rearrangement of these helices, which ultimately brings the two transmembrane domains closer to each other. It also allowed us to see how this nanobody acts as a positive allosteric modulator. So with those changes in the fly traps, the agonist binding domain, now we'll focus on the transmembranes. And you can see in the inactive state, these are well separated. There's no contact between the two protomers. But in the active state, we have this interaction between transmembrane 6. And this just again shows the kinds of rearrangements that have to occur, very large scale rearrangements. So we had a question as how important this interaction was. We developed a disulfide bond between the two protomers, expressed them, and found that this is an activating state. Black is the wild type, and the blue is just the control. Our two cysteine mutations that we placed were both constitutively active, so they're completely active all the time, and there's no further activation with glutamate. So simply forcing this interaction is enough to lead to activation of the receptor. Now, how is the signal transmitted from the fly traps to the transmembrane domains? Well, you can see it looks like a fairly flexible, weak interface. But if you focus on this loop, which is between two of the transmembrane domains, and this is called the cysteine-rich domain, there is a there's a weak connection between this extracellular loop 2 and the cysteine-rich domain. And this would be sufficient to enable the cysteine-rich domains to sort of push the two transmembrane domains closer to each other. So to test this, we mutated, or we deleted the tip of this loop, just making it a little shorter. And it led to complete inactivation of the receptor, even though it's still bound agonists. So that's sort of the end of my tour of cryo-EM and its impact, its huge impact on the GPCR field. Now we largely will only use cryo-EM when going after active state structures. Inactive states without the associated G protein are still a challenge because they're relatively small size and the micelle. However, with tricks of protein engineering and antibody fragments, it's looking more likely that we'll also be able to get inactive states. And this is, again, a really important advance for drug discovery because pharmaceutical companies can now rather quickly identify the location and interactions of their favorite agonist or modulators to their GPCR targets. So I'll end here and take any questions. These are my colleagues and collaborators, particularly acknowledge Bill Weiss, who worked with us on all our crystallography projects, and Yorgos Kiniotis in his lab on the cryo-electron microscopy projects.
Great, Brian, that has been a fantastic talk that gives us a, a tour on this family of proteins. So there are a couple of questions from the audience. The first question is, do you have any thoughts on why such non-canonical states are not observed for any other class A or B G protein coupled receptor complexes? Do you think that has to do with the sample preparations or other biochemical factors?
So that's a good question. We are now starting to see in a few other receptors non-canonical conformations. They haven't been published yet, but they're not exactly the same as our non-canonical conformation here. One possible explanation is that this particular receptor, the neurotensin receptor, as I mentioned, is promiscuous. It couples to literally all major families of G proteins, and maybe it's related to that, although I still can't explain why it would be related. But we were, I think, just lucky to be able to capture it in the act of selecting a particular type of G protein.
Great, thank you. There's a question from Dr. David DeRosier. The question is the inherent flexibility of the proteins. Do you think that is the limiting factors to prevent it to go beyond 3 or 4 angstrom in cryo-EM structure analysis?
I think it's definitely one of the limiting factors, although you know, with improved data processing and particularly if you have high affinity agonists, the G proteins also sort of prevent and stabilize some of this flexibility, that now some of the receptor G protein complex structures are going below 3 angstroms.
Great. And there's a question from Dr. Ganjand. The question is that they are working on another protein called adenosine A2A receptor, and they are trying to solve its dimeric structures, and the food length of the single polypeptide is 40 kilodaltons, so the dimer is 80 kilodaltons. So what's your opinion about the prospect of solving this structure using a cryo-EM?
Well, I think it increases the chances of being able to get a structure because it will be larger and it will have a more oval shape. but it's still going to be, you're still going to have some problems with the micelle. You might end up trying a number of different detergents to try to reduce the size of the micelle, but it would definitely be worth a try. If you can form a stable dimer, that would be an interesting structure.
Okay, I have a question for you, Bryan. So since there's so many proteins of this superfamily of the protein coupled receptors, in the case of the pharmaceutical company, how many possible drugs they are working with? Do you think they would have, is there a prospect solving a structure a day so that they can do a big screening in the whole drug discovery pipeline?
I don't think it's well. You know how much microscope time is needed. It's definitely possible to solve structures in a rather rapid rate. So with new data collection strategies, you can probably collect enough data in maybe even 24 hours if you have access. Then of course it depends on how quickly your data transfer and how much data processing bandwidth you have. But I would say everything being ideal, you might be able to get a structure within a week, particularly if it's an iterative structure. So if it's a protein that you've solved before, and it's just with a different drug, model building and so forth will be much faster. So it can be a very rapid turnaround. We've been able to. My wife has a small biotech company that does structural work for pharmaceutical companies, and we've been able to get our company fairly rapid turnaround on their discovery projects, and not within a couple of days, but within a month or two, and that's definitely helped them in terms of making decisions about how to modify the compound.
Good, thank you. There's a question from Lester Saluzet, and he enjoyed your lectures, and he wanted to get a sense of the size of the displacements that you talk about in terms of the movement
in terms of, you mean like the size of transmembrane 6 displacement?
Yeah.
Yeah. So that's the biggest change in most GPCRs, and it can be as much as 12 to 14 angstroms. For family B, it's even larger. I think it's about 16 angstroms. But the others are more subtle.
Yeah. So there's another question from David DeRosier. The question is, do you understand why the detergents and lipids are necessary for the prep, particularly when you synthesize?
Why the detergents and lipids are necessary?
Yeah.
Well, so these are native membrane proteins, and the outside of the protein is largely hydrophobic. So it wouldn't be soluble in an aqueous solution without detergents. Many of these also really are cholesterol binding proteins as well, and many GPCRs require cholesterol to be fully stable. So even if we're using detergents, we use a mixture of detergent and cholesterol.
Have you tried to use a nano disc as a substitute?
Yeah, so the structure I showed you of the inactive state of the family C was actually obtained in a nanodisc. If you don't use the nanodisc, you have these two detergent micelles sort of separate out from each other. So you need actually the nanodisc in order to keep the complex together.
Okay, good. So another question from Dr. Yi. What do you estimate the percentage of the intermediate conformations in your membranes? Can we assume that you can capture a large numbers of different conformations, or there's a limited conformation you can capture?
So the most information we have comes from muscarinic receptors, where we did NMR studies, and we can see each agonist, each structurally distinct agonist stabilizes a distinct conformation. I mean, some are, they're probably not large changes, but they're clearly detectable changes in chemical shifts. So I think they're probably a limited number, but it's not 2 or 3. It's more like 5 or more.
I see. Good. All right. There's one last question from Dr. Saxena, and the question is that possible to use the cryo-electron tomography to capture the in vivo structure of the GPCR to high resolution?
I don't know. I hope so at some time. I think there are other people listening that could better answer that question than I can.
Okay.
That would be very exciting.
Yeah, good. All right, good. I think I'd like to thank Bryan for this very stimulating talk, and that's a great tour that you provide us. Thank you, Bryan. There's some more questions from the audience. If you are able to stay around at the end of the next lectures, and then there may be even more questions from the audience. Thank you.
Okay.
Thank you, Bryan. Wonderful introduction to the field. Thank you. So we'll now pass on to the second speaker, and I'll ask Jen Dion to make the formal introduction. I'd just like to say that in addition to many contributions to research and in situ microscopy, Professor Olson has also been very active in the International Electron Microscope community. She is currently General Secretary of the International Federation, and if the normal line of succession happens, then she will have even more responsibility in the future, for which we all truly appreciate the efforts that she's put into this. So Jen, please.
Great, thank you so much, Bob. It's my pleasure to introduce Professor Eva Olson, who is a professor of experimental physics at Chalmers University of Technology in Gothenburg, Sweden. As many of you know, Eva is a leader in the field of electron microscopy, with research focused on the correlation between the atomic structure and properties, including the transport of charge and matter and the effects induced by electric field, light, mechanical strain, and temperature. Before starting her career at Chalmers, she received her master's degree in engineering physics at Chalmers and also her PhD in material science in the Department of Experimental Physics, also at Chalmers. And then she conducted a postdoctoral fellowship at the IBM Thomas Watson Research Center in Yorktown Heights. As Bob mentioned, she's presently serving as the General Secretary of the International Federation of Microscopy Societies, or IFSM, and she's also served as a General Secretary and the President of Scandem, which is the Nordic Microscopy Society. So she's had tremendous service in the community. She's also chaired several research committees and evaluation panels, including the Engineering Physics Evaluation Panel of the Swedish Research Council and the Research Committee of the Swedish National Space Board. And among many honors, in 2011, she was an elected member of the Royal Swedish Academy of Sciences, the physics class. And in 2017, she was also a fellow of the Japan Society for Promotion of Science, hosted by the University of Tokyo. Eva, it's such a pleasure to have you here. And without further ado, I'll hand over the floor to you.
So thank you very much for the introduction and also for inviting me. It's really an honor to be here and to share some of our results on in-situ electron microscopy. So I hope that you can see my presentation.
Yes, it's perfect.
And so the topic is then in-situ transmission electron microscopy. And the focus will be on piezo-driven manipulation, probing, and perturbation in the microscope. So then, first of all, what is the definition then of in-Situ electron microscopy? Well, according to Wikipedia, it is an investigatory technique where an electron microscope is used to watch a sample's response to stimulus in real time. And we can then observe many processes in the electron microscope. So we have growth processes, melting, reactions, liquids, electronic properties, mechanical properties, magnetic properties, optical and thermal properties. And today then I'm going to focus on the use of in-situ manipulators to perform studies of the properties within the microscope and get the site-specific information. And in particular, I'm going to show the use of a scanning tunneling microscope inserted into the TEM holder. And it's a TEM holder is schematically shown here. And the part that is interesting now is up here, where we mount the sample here. And then we have a piezo tube that is giving us then the fine motion in X, Y, and Z direction. But then in addition, which is very nice for this particular type of holder, is that we also have this mechanism. So that means that we are giving a wide dynamic range for the spatial motion of this tip that is our manipulator. So we can move this tip in the range of millimeters and then down to the sub-ångstrom dimension. And this is an example where we have an STM image of a graphite surface obtained in the TEM. So we are able to actually do the STM imaging within the transmission electron microscope using that function. But then if we start with the manipulation part of the work, so we can then use the STM tip, which is a gold tip in this case, and grab a silicon nanowire that is 40 nanometer in diameter, and we can bend it while observing in the TEM mode. And we can repeat the experiments, and we can also increase the strain so that we get fracture in the microscope. And while we're doing this, we can also measure the electrical transport properties. We can study electromigration, and we can use that as manipulation. And we can also do deposition within the microscope. So this is now the illustration of the manipulation on the fine scale while we are observing in the microscope. Another nice feature is that if we now take an example of a carbon nanotube, and we can see a fairly long carbon nanotube here, and what we want to do now is to use this manipulator and make contact at different positions along the carbon nanotube. And we can understand that it's easy to reach these two positions, but the question is, what do we do with these positions out here on the side, having this geometry of this gold wire? Well, what we do is that we take this gold wire here using the millimeter ability to move it and take it over here on this sample and then simply bend it. And then we are able to reach also on the side with the very same shape of the manipulator. So it is a very nice feature where we can actually use a joystick where we move around our STM tip here in the microscope to access the sample and also do the manipulation. And this is another example where we're now getting into the probing. So we use the gold, in this case, to make contact to a gallium arsenide nanowire. And we can bend the nanowire in the microscope, and we can measure the IV characteristics as a function of the bending and the strain that we then have in the nanowire. So this is now part of the probing that we can perform in the microscope. And what is always important to remember when we perform the probing in the microscope is to make sure that what we are measuring is actually something that is representative for the structure. And when we are performing electrical measurements in the microscope, well, then we need to make sure that we actually have a very good contact between our specimen, in this case, a gallium arsenide nanowire, and the contact and the manipulator that we then make in contact with the nanowire. So these are then measurements showing how we have optimized the electrical contact between the STM tip and the gallium arsenide nanowire. And this is another example, and in this case, it's a solar cell type of material, copper indium gallium selenide structure. We have our STM probe, and we have then made current paths through the structure, and we are measuring the Ivy curve. And we are also then looking at the effect of the size of these cross-sectional areas to make sure that we have a proper scaling of our electrical measurements. So control experiments are very important, and we always need to remember them. So the type of materials that we are studying doing the in-situ microscopy are then materials for energy technology. We have materials for quantum technology, and we also have soft materials for health and energy that we are studying. And as mentioned in the beginning, we are studying the effect of electric field, temperature, light, and also strain on the properties as well as on the atomic structure. The main instruments that we have used for the results that I'm now going to show are then a Titan microscope that can be operated between 80 and 300 kV. And it's also equipped with a beam filter as a monochromator. And we can perform EELs with an energy resolution of 45 millielectron volt. We also have a dual microscope, and here we can operate down to 40 kV, and we can have an energy resolution of 20 millielectron volt for the EELs spectroscopy. So let's then move to a case where we use perturbation, that is to say stimulation within the electron microscope. And this is now an example where we're going to study gold, and we will use gold nanocones. And in this image, you see the high resolution TEM image of this gold nanocone. And we are going to study an electric field induced structural change. And the main scientist on this work is then Ludwig de Knoop, and you see the reference for the work down here. So we are going then to, first of all, look at the sample preparation of these gold nanocones. And then we're going to see how we can do the nanomanipulation in the microscope. We are going to look at atoms and how they are moving in the microscope. We are going to check the temperature increase due to the effect of the electron beam. And then we're going to look at cold field emission, field evaporation, and also see a structural switch caused by the applied electric field. So the gold nanocones, they are made using a whole mask colloidal lithography method. And we are then putting the gold nanocones on an amorphous carbon film, and the film is floated off in water. And we extract that carbon film with the gold nanocones using a gold nanowire that has been mechanically cut. And here we are looking at an SEM image of such a gold nanowire. We have it here. And then if we go magnify this area, we are starting to see some details. And then eventually, if we go even further up in magnification, we see these bright dots in the SEM image, and these are then the individual gold nanocones. So this is now a schematic of the in-situ manipulator holder. So we have the STM tip, and we have the sample that is mounted here. We see then schematically the carbon nanofilm with the gold nanocones. And we can look at them in the TEM. So we have the gold wire, we have the carbon film, and we have the gold nanocones protruding from that amorphous carbon film. And if we then go further up in magnification and do a high resolution TEM imaging, then we see that this is a single crystal tip of that gold nanocone. We can see that the dimension here is of the order of 5 nanometer at the tip, and it's about 180 nanometer in length. And then we can apply an electric field between the STM tip and the sample here. And because of the shape of this gold nanocone with a very sharp tip, that means that we get an electric field enhancement, so that we get a very strong electric field exactly at the tip of this gold nanocone. And this is illustrated then in the schematic, where you see the color scheme showing then the enhanced electric field. So the first step is now we want to have 2 gold nanocones facing each other. And then we use the nanomanipulator function. So we have then the STM tip that we are moving around, and we are extracting gold nanocones from this amorphous carbon film up here. And now we are getting to the point where we have 1 gold nanocone that we can get in the geometry, the orientation that we desire for our experiment. And slowly, step by step, we are extracting that gold nanocone. And we are moving to another part of the carbon film where we have another gold nanocone protruding. And now we apply a small electric field in order to align the 2 gold nanocones so they are exactly facing opposite each other. So now we are ready for performing the experiment, where we now are going to apply an electric field on this gold nanocone. So the original purpose was actually to observe cold field emission of electrons from this gold nanocone. And in this graph, we are looking at the number of atomic layers that we have on this gold nanocone as a function of time. And we have different colors here. So we have the blue, which is no bias applied on between the STM tip and the cone. The red one is no bias again, but now we have removed the electron beam from the specimen. And then the third green one is when we both have bias and the electron beam on the tip. So when we look at this, when we have the electron beam on the gold nanocone, we see that we remove atomic layers from the very tip, the very apex of this gold nanocone. So you have the red markers here, they are reference marks, and we see that we get a shorter nanocone. So we wanted to understand what the effect was of the electron beam. So this is when we then removed the electron beam. And we then also observed a shortening of the gold nanocone. Now, the interpretation that we make of this is that we do have diffusion of gold atoms, and this is happening at room temperature. And this is commonly observed, and it was even observed by Binning and Rohrer when they were doing the AFM experiments using gold tips. So leaving a very sharp gold tip at room temperature, then you're getting a blunting with time. And what we observe here is that the blunting is 7 times faster with the electron beam on the gold nanocone compared to off. And then if we use the Arrhenius equation, assuming that we have diffusion, then we see then that we have a temperature increase due to the electron beam that is 16 degrees Celsius or Kelvin. But we don't see any change otherwise in the structure of the gold nanocone. It's crystalline, and it's crystalline to the very surface of that gold nanocone. So now we're going to take a closer look at what we have with respect to the structure of these gold nanocones. So in this part that is marked with yellow correspond to these images. And what we see in these images is, again, the crystalline structure of the gold nanocone. We are getting a shortening, and that is again due to the normal diffusion of gold going from the sharp tip up along the surface here. And then we have the red area here, the EFG, and we see the images here. And what is happening here is that here we are now increasing the field, the electric field that we are applying on this gold nanocone. So we went from 0 and now we're going up to 21 volt per nanometer. And so this is actually a very strong electric field that we are applying. And what we see now, if you look in the details here on the structure, we see that we are getting a disordered surface layer on this gold nanocone. And we can also see that we don't see any change any longer. We're not getting a shortening of the gold nanocone in this electric field range. And then we are entering this blue region. And this is where we further increase the electric field. So now we're going from 25 and 28 up to 29 volts per nanometer. And what is happening now is that we are getting field evaporation. Look at the reference marks, and we see a rapid disappearance of the gold atoms. And this is now field evaporations that we are actually observing in the microscope. So now we're going to take a look at this happening. And pay attention to this region in the gold nanocone. And the reason why the image is jumping is because that when we start to increase the electric field, there is a motion. So we are compensated to that. So we're actually looking then exactly what is happening here at the tip. And we are going from the crystalline structure, and now we are entering into the disordered structure. And then we are increasing further the electric field, and we are going into the field evaporation that is starting now. So this is all happening while we are observing. And we are having the atomic resolution in the microscope. So what we actually have seen now is that by applying the electric field, we go from a crystalline structure to a disordered surface structure. And this is all due to the applied electric field. And this switching between ordered and disordered surface layer is reversible. So we can enter into the critical field, get the amorphous layer, and then go down again. And what we do get is, again, the crystalline structure up to the very surface of this gold nanocone. So we are live observing a reversible structural change of this gold nanosurface, and it's a switch. Now I'm going to use a few minutes to also make some comments on thermal measurements in the microscope. So here we have a schematic, and this is now work where we have Norvik Voskanian and also Hanna Nilsson that has performed the main measurements. And we are going to start with a carbon nanotube that is on a silicon nitride substrate. And we are going to use a method where we use indium islands on the backside of a silicon nitride membrane. And we have the carbon nanotube on the top surface. So we are looking here in plan view of the structure. So we have the electrodes, and we can apply a voltage and measure the current in 3 different configurations that are then shown here. We are making use of the fact that indium changes in contrast when it goes from the solid to the liquid. And we can see the effect here if we look in the electron diffractogram, in the diffraction pattern. So in the liquid state, when then we have the rings like this, and the diffuse rings. And in the solid state, well, then we have the more sharp rings that are due to the crystalline reflections of the indium islands. And if you choose to image in dark field, we can then see that we have more electrons entering into the aperture for the liquid phase compared to the solid phase. And this means that when we look into this dark field image, we understand that these indium islands here, they are in the liquid state. Then we have an isotherm of 156 Kelvin. And then we go into, sorry, it's Celsius, 156 Celsius. And then we go into the solid state. A quick illustration of how we do this. So we have the silicon nitride membrane. We deposit gold electrodes, the carbon nanotube that we want to study, the contacts that we deposit using electron beam lithography. We have the plan view, the carbon nanotubes, and the electrodes. We deposit indium on the backside. And now we are ready to do the measurements. And we then can do the biasing. And what we have here are now 3 cases where we have varied the voltage applied to the electrode. So we have a low voltage, and that means a lower current. It's 195 volt. And then what we mark the isotherm 156 degrees Celsius. We increase the voltage, we get another isotherm, and we increase further and we get a 3rd isotherm. And in this way, we can actually observe the equilibrium temperature distribution around this carbon nanotube. So I want you to remember now, this is equilibrium. So the temperature stays stable with time once we have reached these conditions. And what we see here is that we are getting a higher temperature at the position of the carbon nanotube, where the current is being drawn through the carbon nanotube. So if we only choose to contact this portion, well, then we are getting the heating only in this portion of the carbon nanotube. And why is that important? Well, the heat conduction through carbon nanotubes is very high. And that means that if we had the heating of the carbon nanotube here, we would have seen not only this portion getting to higher temperature, but also this portion because of the thermal conductivity. So we are actually observing a heating of the substrate, not of the carbon. So we're getting the phonons excited in the substrate and not in the carbon nanotube here in these cases. And that happens up to a certain critical applied voltage and current drawn through the structure. So this was a case when we had the carbon nanotube onto a silicon nitride substrate. What happens if we put graphene on this substrate? So in this case, we now have our contact and we are drawing current between this manipulator here over to the electrode that is positioned down here. So we are drawing current from here down here. And what we're looking at here now is that the voltage is step by step increasing through between this electrode and to this other fixed electrode. And what we see here now is that the entire area here is obtaining the one temperature. So the graphene, because it has a high thermal conductivity, we are getting an even distribution of the temperature compared to the case with the silicon nitride, where because of the poor thermal conductivity, we are only getting this limited heating region. So the take-home messages that I have with my lecture is that, first of all, the in-situ electron microscopy provides site-specific information about the response to stimuli. The piezo-driven in-situ studies allows simultaneous manipulation, probing, and stimulation or perturbation. The information about the site-specific electrical, mechanical, optical, and thermal properties can be extracted and correlated to the atomic structure. The design of the holder that I've shown here, it gives a wide spatial dynamic range from millimeters to sub-angstrom, which is very useful when studying the structures that we insert in the microscope. We have high precision in the manipulation, getting a precision of sub-angstrom. And last but not least, when performing the in-situ experiments, always remember to perform the control experiments so that what you are looking at is actually representative for the structures that you are studying. So I want to thank all the funding agencies that are supporting us for our work. And of course, I want to show our group. And here we actually have, during this time, we had Professor David Smith, who gave the last seminar in February. He was visiting us together with Molly McCartney from Arizona State University. So with this, I thank you for your attention.
Thank you very much, Eva. This was an absolutely wonderful talk. There are a number of questions in the chat or the Q&A. If any audience members has additional questions, feel free to enter them in, and I will try to address them to Eva. Of course, you ended your talk by mentioning the importance of control experiments, and whenever anyone is doing in-situ electron microscopy, it's important to consider the effects of the beam. Do you find that for different materials and different measurements, you're doing different control experiments to control for the beam? Could you talk a little bit about avoiding beam effects?
Yes. So the electron beam is definitely something to consider and also to keep in mind. And what we have seen is that it's very hard to predict what is going to happen as a result of the electron beam. So each time that we start a new experiment, we really need to find out what the mechanism, if there is a damage effect of the electron beam, and also if there are knock-on damages, or if there is radiolysis, if there is heating, as you saw in this example that I showed with the gold nanocones, or if there are charging effects, for instance. So we are actually working now on zeolites, and what we see there is that we do have an effect of the electron beam. So we are starting with quantifying the effect of the electron beam and also how it relates to the electron beam dose and also dose rate. We are also exploring the use of graphene, because there are some reports in the literature where graphene can actually reduce the effect of the electron beam. And we want to understand if it does reduce the effect and also why. So I think that my advice is always check in the beginning of your experiments, do you have an effect of the electron beam? Is it affecting the information that you want to extract from your samples, well then there are ways to reduce the effect of the electron beam or to get a window where you actually can perform your experiments.
Great. Yeah, absolutely. I'm excited to hear how the graphene measurements unfold. It would be exciting if that could mitigate the effects.
I am too, yes.
Another question is about your beautiful experiments on the gold nanocones, where you looked at the change in crystallinity with applied electric field. A question from the audience member is if you were able to confirm using either spectroscopy or perhaps even imaging that the disordered structure was in fact gold and not, say, diffusion of some other species.
Right. So in this case, we observed the reversibility on the structure. So, and this is, so once we had formed the disordered surface layer, we could go back down again, and then we came back to the gold structure. So looking at the contrast in the microscope and the structure of the gold nanocone, our conclusion was that this is actually gold atoms and not any contamination on the surface. And in addition, when we observed in the microscope, we didn't see any contamination, because if we have contamination, we can actually observe it very clearly in the microscope.
Great, thank you. Another question was on the excuse me, Arrhenius relationship that you used to determine the beam heating effect, and I thought those measurements were really beautiful. What was the temperature difference that you measured, and were you able to use the lattice reflections to potentially verify that temperature change?
Right. Very nice question. So the temperature increase that we saw due to the electron beam was 16 degrees. And we didn't do this with electron diffraction. And the reason is simply that that would be a very small change in the lattice parameter, the increase of 16 degrees. So what we also see is that depending on the surroundings, so in this case, we had a very good thermal conduction between the gold nanocone and the surrounding. But this also means that we get not such a high temperature increase. But if we would have had isolated gold nanocones, like on the silicon nitride membrane, then we would have had a much higher temperature increase. And we have also observed that in cases where we didn't have that good thermal conductivity surrounding and in connection with the gold nanocone.
Great, thank you. Another question is actually about the detection system that you use. People are wondering, currently, is the frame rate of the camera that you're using a limiting factor in your experiments, and might there be effects at sub-millisecond timescales that would be interesting to directly observe?
Oh, yes. So this was not, yes. So yes, definitely, we are limited by the camera that we are using then for the observation here. So we are now very happy that we are having a one view camera now on our new microscope, but in the future I also want to have the direct detection cameras to make these observations. But at the moment, this was a not so fast and sensitive camera that we were using.
Okay. Well, good motivation for those who develop detectors to keep coming up with new platforms. The final question I'll ask is back to the same experiment. This is kind of like a mini, atom probe microscope that you've set up. And the audience member is wondering whether the evaporated species is deposited onto the counter electrode. If you make the analogy with, atom probes, you'd expect it to be, basically always the same site.
Yes. So, so this is, as you say, this is an analogy to atom probe microscopy. And so I can say one dream experiment that we have, and that is to lower the temperature and to perform these experiments at temperatures that are used for atom probe microscopy to see if we have the same structural change at the tip. This is really very interesting. And then the other experiment that we had in mind and that has not been carried out was that we wanted to actually put like a TEM grid as a counter on the counter electrode to actually collect the gold atoms that were evaporated. So I cannot provide the answer today, but this is something that we are really, that we wish to carry out for future. That is to, because in this time we wish to have that TM actually like a carbon film to collect these gold atoms and then do the analysis on that grid, but we didn't have it yet.
Cool. Sounds like a really great measurement. Also in these experiments, were you able to do any spectroscopy to map out plasmonic excitations that might be emerging, from irradiation, even just with the electron beam? You might be able to get some localized surface plasmon resonances that could influence, the migration of the atoms or the local temperature.
We have actually done preliminary measurements where we have been looking at the plasmons as a function of distance between the gold tips and also as a function of applied electric field. Now, the thing is that it's the density is very low. And so this, at least until now, these experiments are inconclusive. So that is work for future.
Okay, well, I look forward to seeing those as well. I think I've gotten through most of my questions and many of the ones in the chat. So I will hand it back over to Bob. Thank you very much, Eva.
Thank you.
Thanks, Jen. And thank you, Eva. Wonderful talk. I think one of the things which struck me was something which is rather underappreciated by the continuous recording of in-situ microscopy, whereby you can see everything that's happening. And so you can put a fiducial mark on, for instance, at the atomic level. So you know which is the plane of atoms which you're looking at, and then which one is then evaporated gradually. And this is an underappreciated advantage of the continuous in-situ recording, that you can put those marks on for the recording at the atomic level. So that's just struck me in addition to the wonderful experiments that you described.
Thank you, Bob.
So thank you again, and thank you, Bryan. We will draw this to a close formally, and we'll stay online for a few more minutes for people to